Alrighty, so we are going to talk about how to do DNA mapping. So this is for a plasmid. Our plasmid that we're starting with has 2,686 nucleotides. And we are so up here we have our restriction enzymes. We're going to call this one A. And this one is B. And this one over here has both of them. So we are going to have to look at both to do this. So when you're working with restriction enzymes, this map shows a digital version of the gels that you would use, so it's just easier to look at. And the first thing you have to do is look at the length of these pieces. So this piece up here and this piece down here are where A has cut the DNA, and the two pieces are going to add up to this. But to know how long the piece is, just drag across, and that's roughly 320. And we're going to ignore these top pieces. So if you drag this one across, that's roughly 220. You're not going to get it perfect. Sorry, I'm doing this with a mouse, so my handwriting's not the best. And then this one carry over is about 900. So what else you need to notice is that this plasmid here, or this DNA chain, is the exact same as this one here. So these two are the same, which means that this one here in both was only cut by A. So how you're going to map it is you're going to make a cut just right at the top for A, and then you're going to go over 320 nucleotides. Just going to take you about here and make another cut for A. So what you'll notice too is that this one here is the same as this one here, meaning this one is made only by B. So these two up here are made by A and B. So we know the length is 900 one. It lined up right there. So we're going to plot it first. We're going to do this one right here. Is made between an A slice and a B slice. So we're going to go from our A slice, we're going to go over 900, and it's going to take us to here. So that's where our B slice is going to go, our first slice. So then we know that a 220 nucleotide chain here was going to carry up. So 220 takes us to about here. And this remaining length, we'll do in purple, is this right here, just goes right here. And it should add up perfectly to this number in the end with your 4 pieces. And you know your A, you know your B. And that's what you need to map. And then when you're doing it, you're going to have to put your numbers on. So you're going to want to, right here, you're going to want your 2,686, and then divide by 2 to get the quarters, divide by 2 to get the eighths. And that should be enough numbers. And if you want, how I did mine on paper was I put my numbers on all of these ones. And then if we're going to go over here, I just did dashes to show where the 16ths would be. But you don't have to do that, as long as you know that, here I'm going to erase some of this so I can show. As long as you can see that this lines up with this, meaning this piece here was made only by A's, and that this piece here lines up here. So this piece was made only by B's. And that these two pieces up here, they don't line up anywhere. They don't line up with the rest, meaning that they were made from both, and you can plot it based on that. Start with your A piece, know that one of these gap pieces goes in between, then you have your B piece, and then you have your gap piece. You know your structure, and then all you need to know is the numbers that follow. So this, of course, isn't going to make a whole lot of sense unless you've also done your biology class leading up to this, but I hope that this is pretty clear and that you have a better idea of how to do this.